kind of simplify or oversimplify, and the attributes, oh, male versus female, etc. And yet generally you can see that you have quite a lot of female specific problems, so ovulatory failure, tubal damage, endometriosis, or other issues with the cervical mucosa. And then of course you have other minor issues as well, which count for about 10%. A very large chunk of the pie, though, can be attributed to sperm issues, so issues with male fertility. Now, if you look at just these figures, one would ascertain that perhaps, okay, well, you could say that it's about 50-50, and that's what most textbooks and most definitions will tell you, that it's a 50-50 issue, 50% female issues, 50% male issues. However, that's not entirely true, you see, because there's an even bigger piece of the pie over here, which is unexplained. So for all intents and purposes, we know that something's wrong, but we can't really explain what is wrong. And a very large section of this unexplained piece of the pie is actually more attributed to sperm issues. It's more likely that there's a sperm issue involved rather than a female issue. So if you put everything together, really the vast majority of reasons underlying infertility is a sperm issue. So, girls, if anyone brings this up with you, you have my permission to basically attack them and say, no, it's a male issue predominantly, not just a female issue. So girl power, as far as I'm concerned on that one. So because of, these multifaceted reasons and a number of other issues as well, infertility, is increasingly affecting a number of people's lives. In fact, it's become such big of an issue that in 2010, the WHO actually recognized infertility as a global disease. Okay, so it's an actual disease that is recognized. And partly due to this increasing urgency, a number of technologies have been made to try and rectify these cases. Okay, now it's not just one technology. In vitro fertilization, or IVF, is a very big umbrella term, and there are multiple techniques that are involved. And collectively, these are known as assisted reproductive technologies. And collectively, they're pretty good. They can treat most cases, about 70% cases of infertility. Now, you have to be careful here, because saying 70% of cases doesn't necessarily mean this will lead to a successful treatment. This just means that ART is applicable or can be applied to 70% of cases. Having said that, it's been quite successful since its first inception, way back when, in the late 60s, early 70s. Since then, more than 4 to 5 million babies have been born worldwide. Actually, these figures are a little bit old. The number is much, much more than that. Now, it's so widely used, ART, that actually in some Western countries, in Europe particularly, I won't name the countries, but 7% of the total birth rate is accounted for by ART. Now imagine that. It's a completely mind-blowing figure, that 7% of entire countries' populations are beginning life in a laboratory, essentially. So that's the kind of background. Okay, infertility is an issue. There are lots of stuff is being done to try and rectify it in terms of treatment, and it's relatively quite successful. But before we can understand what you actually need for ART, let's go back a little bit further. Let's go back and refresh a little bit in terms of our background. So what exactly is it that you need to create life? Now, of course, under normal settings, this would have been a very interactive part where I would have asked you and you would have suggested some questions. I would have made fun of you, but that's not going to happen for this particular format. So we'll just kind of go straight into it. Now, the reason I'm asking this particular question is, well, traditionally, in terms of science at least, we haven't really fully realized in the past what exactly it is that we need. Now, of course, we know at the moment that we need a female component and we need a male component. But of course, humans are really funny creatures. We tend to overgeneralize, and our overgeneralizations always tend to be quite complex in many ways. And to just illustrate this, I'm just going to go through a brief history as to what ART is and where it comes from. So just relax. You wouldn't really be expected to know all of this for your exams. This is just really for pure interest and to set a background for you. So the first experiment that was done, which I'm going to talk to you about, is something I like to call frogs in pants. Okay, and this was done by this gentleman here, this chap called Lazzaro Spallanzani, Italian dude. And what he did, because he basically had way too much time on his hands, he took frogs, male frogs specifically, and divided them into two groups. And in one group, he just left them completely as they were. And on the other group, these poor frogs, he made them wear these specially designed rubber pants. Okay. Yeah, he had way too much time on his hands. He specifically designed and made these pants for these frogs. And what he then did was unleashed both groups of frogs on females. And of course, what he found was that frogs without any pants, they were able to result in tadpoles. Whereas the frogs with the pants, they of course weren't able to result in anything. Now, apart from demonstrating that basically this guy was basically a pervert with way too much time on his hands, but for the first time, really, Lazzaro Spallanzani showed that there was some sort of male involvement or male component that was involved. Of course, now we can say that this was the first proof that that established that sperm was required. But of course, this was way back when, and being humans, we kind of tend to take things to the extreme, as I mentioned. And apart from these sort of experiments, what a lot of people also don't realize is that when the very first microscopes were being made, and there's a reason I'm touching upon microscopes, because this field, ART, is without microscopes, you can't really do anything. So it's integral. So it's only fitting that I mention the history of this as well. That one of the first things that were mentioned when microscopes were first made, the first thing was pond scum, so pond water. The second thing was, of course, human sperm. And there's actually a really funny story behind this as well. It was actually, these observations were first made by a Dutch scientist called Anton van Leeuwenhoek. He was the apprentice of another person, this chap called Hartsucker. But essentially, before I go into the story, what these individuals or what these scientists suggested, well, after looking at sperm under a microscope for the very first time, was something called the homunculus theory. And what this suggested was that in the sperm head, you had the individual, the new baby. So essentially, the sperm head was the thing that was transporting the new individual to the female. And this was later modified into what's known as the animalcule theory, which basically stated that the sperm was a miniature version of the animal that produced it to begin with. And, you know, in some aspects, it's actually quite amazing how accurate these early microscopes were, because yeah, you know, it's a bit of a stretch to say that you have babies inside the head of the sperm, but you know, considering the primitive type of technology that was there then, it's actually, if you look at the morphological features of sperm, you can actually kind of see where they were coming from. So of course, you know, we know that this isn't actually the case, that, you know, you don't actually have babies inside sperm heads, or the sperm isn't a miniature version of the individual itself. But there seemed to be a theme in terms of these assertions, and these assertions were that basically you needed the male component only. You didn't need any female involvement. So of course, we know now that this isn't necessarily the case, but at the time, this was taken to be a science fact. And then you move a little bit further down in history, and you find that actually, you know, going from only a male component with no female components required, you know, humanity just kind of turned its head completely. And this was done by this chap over here called Jacques Loeb. And at the turn of the 19th century, around about 1899 to about 1901, something like that, this chap basically took sea urchin eggs and he changed the salt concentration of the water in which they were in. So he basically added or decreased the amount of salt. And what he found was doing this caused these sea urchin eggs to start developing into embryos. They started undergoing embryogenesis without any sperm required. Now, I shouldn't say doctor at the time, it was Mr. Loeb, but Jacques, he had essentially stumbled upon a phenomenon that we have really well characterized now called parthenogenesis. But at the time, you went from essentially saying that, oh, sperm is the only thing that's required, to an experiment showing you that actually sperm isn't required at all. Now, of course, these embryos, they won't develop to term, they'll develop to a special stage called the blastocyst. But, you know, they won't develop any further because they don't have the proper genetic components. But at the time, this is quite astounding. And it creates a lot of press, of course, as well. But this is just to demonstrate to you how we've oscillated from one extreme to the other. But thankfully now, drawing our history lesson to a close, we know that actually it's more or less an equal involvement. Both gametes are just as equally important as another, okay, in terms of sexual reproduction per se. You need a sperm and you need an oocyte as well. So we know that you need male gametes and you need female gametes as well. So assisted reproductive technology, all it's about really is to try and bring the two together in an optimal condition to create a novel individual or to initiate embryogenesis. Now, when I say ART or assisted reproductive technologies, we tend to think this space age type of laboratory with people in hazmat suits and sterile environments, etc. But ART has actually been around for quite a long time. Okay, now if you think about it, we've been doing ART ever since we've tamed animals. So in terms of selective breeding, that's a form of ART. Artificial insemination, that's ART. We've been doing it for our cows, for our sheep, even for our dogs. In fact, the very first artificial insemination, very first technically ART, was reported in dogs. This was early as 1783. And in fact, the modern application of assisted reproductive technologies, or IVF, in vitro fertilization, as we know. This was really more in the 1960s, early 1960s, late 1950s. And this was done in rabbits. And this was done by this chap here, called now Professor Sir Robert Edwards. Okay, and this was also the chap who was responsible for the very first human IVF. However, he wasn't alone in this. So Sir Robert Edwards was actually performing these IVF experiments within animals, rabbits being predominantly the case, as well as mice and rats. But the human side of things didn't really come around until Rob Edwards met with this chap here, American surgeon called Patrick Steptoe. Now, Patrick Steptoe at the time, he was pioneering a form of surgery called laparoscopy. And specifically, it was for a specific reason. Now, of course, we know this as in lay language keyhole surgery. And at the time, Patrick Steptoe was trying to apply laparoscopy for essentially tubal surgery. Okay, so he was trying to utilize laparoscopic surgery to try and get rid of tubal blockages at the time. Now, in terms of tubal blockages, you can get them at three various stages. You can get them proximally at the mid-segment, or as well as the distal, so near the uterus junction, within the middle of the tube, of the fallopian tube, as well as right next to the ovary itself. Now, surgery, of course, is possible, and even though it's laparoscopic surgery, it still is open air to a certain degree. So any surgery will always increase chances of infection. And even though tubal blockages can be rectified by laparoscopic surgery or similar sort of surgeries, you can always get regrowth of scar tissue. So the scar tissue could just grow again and end up blocking. In fact, it's quite likely that it'll end up blocking at the same place again. Now, tubal blockage surgery, laparoscopic surgery, also seem to be leading to an increased risk of ectopic pregnancy. Okay, so really, through this methodology, it was possible to treat tubal blockages, but it wasn't really ideal. Okay, so combining forces, both Robert Edwards as well as Patrick Steptoe, they performed the very first human IVF, where the IVF technology pioneered by Robert Edwards was utilized to create embryos, and then the laparoscopic surgery pioneered by Patrick Steptoe was used to reintroduce the embryos back into the uterus. And this led to the very first IVF baby over here. It's called Louise Brown. That's her name. And, this is a before and after pic over here. So this is actually quite recent, as I believe 2008, 2009. And this picture is actually quite meaningful as well, because Louise, who's all grown up in this picture, is also pregnant with her first child. It's a very, very poignant picture. And really, this picture is also quite poignant because it was taken shortly before Sir Robert Edwards passed away. And it was quite sad, actually, because for all of his efforts, he wasn't actually recognized until very recently. IVF in vitro fertilization and associated technologies won the Nobel Prize for medicine. I believe 2010, 2011. And it was even more unfortunate that by the time the award was made, Robert Edwards had unfortunately passed away. So it was his widow who accepted the award. So it's a Nobel Prize winning field, and it really has revolutionized not just fertility treatment, but all sorts of biomedical technologies as well. So that's a brief history. Now let's quickly jump into the principles of ART proper, into what exactly would ART involve. And really, you can split ART into 3 specific stages. Okay, the first stage here would be gamete isolation. So you'd have to obtain the sperm as well as the egg or the oocyte. The second stage would be combining these to generate an embryo. And then the third and final stage would be choosing a good embryo and transferring it back into the uterus. Okay, so basically, in a nutshell, this is what ART is: getting gametes, combining the gametes, getting the embryo, and transferring it back. So in terms of gamete collection, this is essentially what you need. Okay, you need lots of sperm, of course, and you also need an oocyte. Okay, so here you have your oocyte here, you see the polar body over there, and then the surrounding zone of pellucida. So this is an actual human oocyte as well as human sperm. Now, any hot-blooded male will be able to basically tell you that more or less getting sperm, unless there's a specific male factor condition that's involved, is actually relatively pretty easy. You know, you should be able to get tons and tons of cells in terms of male gametes, and then there are various techniques used to purify these and get the best quality samples. But the issue really comes most of the time when you're trying to isolate the oocytes. Of course, the sperm is produced while following ejaculation, but the oocyte is only really produced following the menstrual cycle, i.e. ovulation. Now, just to remind ourselves of what ovulation is, you're going from primary oocytes and going through oogenesis throughout, maturation essentially, and then you get to the point of ovulation where the mature oocyte is released into the fallopian tubes. Now, usually, of course, if you remember, this is happening in response to various increases and decreases, various cycles of hormones, progesterone, luteinizing hormone, so on and so forth. But of course, most of the time, if you remember from foundation block, most of the time you will only have maybe 1, perhaps 2 oocytes being ovulated at any given time per ovary. And most of the time you only have 1 or 2 oocytes being ovulated anyway. But the whole point of IVF is to try and increase the chances of success. Okay, so essentially you need more than just the 1 or 2 oocytes. You don't want to put all of your, and pardon the pun, you don't want to put all of your eggs in one basket. Okay, so what we want to try and do is to try and increase the amount of oocytes being ovulated. And the way we do that is via hormonal stimulation. Okay, this is usually HCG or GnRH agonist treatments. It's essentially pumping the poor female full of hormones. And the way in which hormonal stimulation can occur would be an intramuscular injection. Could also be a subcutaneous injection, so just in the first layer of skin. And you also have other methodologies such as nasal inhalation over here. The more common one at the moment is subcutaneous injection. It's self-administered. And you know, this is essentially results in a number of oocytes being ovulated at the same time. After this, after a certain treatment, of course, and this would depend on the actual female, the history of fertility, whether there are any hormonal issues, etc. So each treatment would more or less be personalized. But really, this process after ovulation is a transvaginal surgical procedure. So it basically involves an ultrasound probe attached to an aspiration needle. And what the ultrasound probe is doing is you're basically finding your follicles, and using the needle, you're aspirating the oocytes from the follicles in the ovary itself. So essentially what you're getting is you're getting multiple oocytes being ovulated via hormonal treatments. And using this ultrasound probe, you're finding the ovulated follicles, and you're also aspirating the oocytes from within. So through this way, you're able to get a number of sperm and hopefully a number of oocytes as well. So usually you'd be getting around about, in a very good case scenario, you'd be getting anywhere from 8 to 15 oocytes, and that's usually a good number. But of course, it's not just about number as well. Number is a good place to start. The higher the number that you have, the better your chances of success. But you don't want, again, to put all of your eggs in one basket. Again, pardon the pun. You need to be able to maximize the amounts, because remember, this isn't a very straightforward process. There are multiple stages which are involved, and just the hormonal treatments in females can actually be very, very painful. For all the women out there, you completely understand when I say that, the menstrual cycle itself, just the natural cycles of hormones is uncomfortable, to say the least. Imagine you're being pumped full of even more hormones. Imagine how painful and how uncomfortable that will be. So it's not ideal to do this again and again and again. Okay. At the same time, there's also financial implications. There's also, of course, the emotional and stress and duress that's involved. So you want some other criteria to try and determine which of these gametes are of the best quality. Okay. And of course, throughout various experiments, a number of criteria have been established, and it's fitting as well that since microscopes are really the only tool that we have to make these judgments, most of these criteria are based on morphological criteria or shapes or how they look. So basically, it's a beauty contest. Okay. Of course, humanity being quite shallow as we are. So for oocytes, the criteria involve the thickness and the number of layers of the cumulus cells that are surrounding the oocyte, the thickness and the size of the zona pellucida, the morphology of the oocyte itself. Does it look nice and round like this beautiful picture over here? How does the polar body look? As well as the clarity of the cytoplasm. Okay, so purely morphological considerations. So in terms of sperm, you also have morphological considerations. So the type of the head, the shape of the head rather, the shape of the midpiece, the shape of the tail, the clarity of the cytoplasm as well. Do you have any vacuoles? Do you have any imperfections within the cytoplasm? But then you also have additional criteria which is specific to sperm. So you also have the concentration. So you have sperm count. This should be above a certain number. This indicates normal spermatogenesis. The motility should be, of course, within a certain range. Motility meaning the ability to swim. This needs to be in a certain range as well, because, well, the sperm needs to be able to swim to be able to reach the oocyte. Of course, the morphological differences we suggested, the morphological criteria we just talked about. Also, we need to look at, specific to sperm, something called viability. Now, this is usually done by the use of dyes, and the viability basically means whether or not you have perforations or holes within the plasma membrane. Okay, so if dye is able to go inside, that means that the membrane is perforated, it's got holes in it, it's not very good. So on top of that, there are also a lot of how should I say, experimental, quote unquote, technologies that are being used at the moment. And one of these is also increasingly, which is gaining attention, something called DNA damage. Now, of course, we know that all cells of DNA, you also should know by now that within the sperm, you have a very highly packaged, highly condensed DNA packed into the sperm head. And sometimes this packaging process isn't very efficient. Okay, you will sometimes get results in damaged sperm, and this technique is basically looking at how damaged the DNA is within the sperm. If you have more damage, you won't be able to get normal embryogenesis. So, multiple criteria which are being established, but the majority of these seem to be purely or largely morphological. So, once one has obtained the gametes, and once one has identified and isolated the best quality of these gametes, we then need to be able to choose what type of methodology we're going to use. Now, for this section, of course, there's many different types of ARTs that are being used out there. I'm going to only cover the major three which are considered to begin with. And of course, even these major three, they're much larger umbrellas for even more specialized technologies in the middle. So the very first one is intrauterine insemination, or IUI. Okay, that's something that we'll talk about first. Going on from that, you have in vitro fertilization. Now, this is something you have to be very careful. Technically, all of ART is in vitro fertilization. In vitro means in a dish. So all of this is happening apart from, of course, IUI or in similar technologies. Most of these technologies are occurring within a dish anyway. But when we say IVF in the context of a specific technique, this is something different, okay? It's a more specialized technology. And of course, you have something called intracytoplasmic sperm injection. Now, IUI, to begin with, intrauterine insemination, is essentially, you would say, a minimally invasive technique, let's say. This is where sperm is inserted into an injection straw. The straw is then inserted into the uterine cavity, and the sperm is then released within the uterine cavity. Now, this is usually involved in cases of sperm donation. It's perhaps also involved in cases of, say severe physiological dysfunction at the vagina or the vaginal uteral complex over here, the junction rather, not complex. And most of the time, it's something that is quite highly successful, especially in terms of sperm donations. It's used quite a lot. But there are, of course, certain risks that are associated with it. So sperm needs to, of course, to be able to be used for this particular methodology, sperm needs to be, of course, a certain concentration. It needs to be of a certain motility, as well as a certain morphology. Because really what you're doing is that you're relying on the ability of the sperm to be able to swim from the uterus up the fallopian tubes and fertilize the sperm over here, about 1/3 away up the ampulla. And of course, it also relies on normal ovulation as well. Now, this technology isn't really that widely used. One would imagine that, this is the one that people would want to use because it's a quote unquote minimally invasive, but there are, of course, associated risks. Now, if you inject too much sperm, then you might be getting something called polyspermy, so more than one sperm fertilizing the oocyte, and this will, of course, lead to failure of embryogenesis. But really the biggest risk associated with this particular technique is ectopic pregnancies, where if the timing is even a little bit off, then you'll get a fertilization event much too early or much too late, earlier being more of the case. And then, of course, you get implantation within the fallopian tube. So again, benefits versus drawbacks as well. So in vitro fertilization as a particular technology, this is the one most basic one that occurs, of course, within the dish. And essentially what we have is you have drops of oocytes. Each drop would contain 1 or 2 oocytes. And then you have smears or streaks of sperm over here. And essentially using a pipette tip, the idea would be after calibration, you would then combine these 2 drops and hope that essentially good quality sperm, a good concentration of sperm, which have good motility and good morphology, they'd be able to swim their way from one drop to another and then fertilize the oocyte. Of course, there are certain criteria which are crucial, crucial for the success. There are factors which are involved. Viability, of course, being one of them. So, if a large number of the sperm are not really viable, the membranes are punctured, or there's a lot of DNA fragmentation, or the morphology is abnormal, then, it won't really matter because the sperm won't be able to fertilize. So you need to have a good proportion of viable gametes. The concentration is also an important factor. If you have too little, then the chances of non-fertilization or unsuccessful fertilization will go up. If you have too many sperm, then you'll be getting something called polyspermy, which is more than one sperm fertilizing the oocyte. And of course, the media in which the cells are bathed are also quite important as well, because there you have different media for different purposes. Sperm require all sorts of different sugars, different ions, and various other factors to be able to be viable. So if you don't use the right sort of media, then you really won't get good fertilization. So those really, most of the time, or at least traditionally, have been the first ports of call. So generally what the way it's been like in IUI would be tried. If that doesn't work, then one would go on to IVF. Of course, that's not always the case. Usually one would try to get ART in the first place because there's something wrong in terms of either a sperm count or sperm issue, most likely that's the case, as well as ovulation issues as well. So the first protocol usually, or should be, I should say, in vitro fertilization. But if that fails, then there is an even more advanced technique that one could use. And this is called intracytoplasmic sperm injection. It's whereby a single sperm is injected directly into the oocyte. Okay, it involves this sort of space age looking technology, and everything you see here, including the operator and as well as the fire extinguisher, is actually vital to performing this technique. So essentially what you need is this specialized sort of microscope, okay? And on either end, you have these two injection tips, okay? You have these two tips. And in the middle, you have your dish with a drop containing your sperm as well as your oocyte. Now, you have two types of pipettes. On the one hand, you have an injection pipette, where essentially you'd be taking a sperm and injecting it directly into the oocyte. You also have a holding pipette over here. Now, this holds on to the oocyte in this way. So what would happen is that your holding pipette, it would hold on to your oocyte so that it's held in place. With your injecting pipette, which has a sharp perforated end over here, you would go into your sperm into a separate drop, isolate the single sperm that you want to use. And then utilizing this, you would inject this directly into the oocyte. And I have a movie of this over here. Hopefully it'll work. Yeah, it's just taking a little bit of time. Oops. There we go. So this is an actual human ICSI being performed. Here you see the sperm, injection being done. And then the oocyte is sucked up a little bit to create a hole in the ulemma and to create a bit of a space for the sperm. And there we go. The sperm is deposited inside and the tip is taken out. Okay, so let's see that again. So I'm going to talk while this is doing, so you can, while this is playing, so you can have a look. Now, this technology is extremely powerful, okay? And it'll get rid of so many different things. You don't need sperm to be motile anymore. You don't need sperm morphology to be completely normal. Even viability, as well as DNA fragmentation, you don't really need these things to be 100% efficient, because it bypasses so many natural barriers, let's say. It's bypassing the zona pellucida, it's bypassing the oocyte plasma membrane. You don't need gamete fusion to occur. So very powerful technique, and it's increasingly becoming the first port of call. So people are just bypassing IVF altogether and going on to ICSI as the first case of treatment. Now, there's some benefits to that. It's much more efficient, but also there's some things which a lot of us in the field are actually quite concerned about. But before we go into that, so you've combined now both gametes, you've selected the best quality, you've combined them using the appropriate technology, and hopefully you should get successful fertilization and successful embryogenesis. Now, once you start getting embryogenesis, you still want to, again, retain quality over quantity. Okay, so you need to be able to decide what is the good quality in this case. So of course, there are different checkpoints, there are different stages that you'd be assessing the embryo all the way from day one. This is actually at the moment a zygote, because the two male and female pronuclei are just combining at this stage. After that, of course, by day 2, you will have 4 cells. And then, of course, you will start to get rapid cell division, as well as creation of specialized morphological structures. By day 4, you have something called, it's this kind of ball of cells, this clump of cells called the morula. And by day 5, you have something called the blastocyst with your inner cell mass over here. You have your blastocele, this cavity in the middle, and then your perfectoderm surrounding the inner cell mass. And by day 6, this blastocyst will then hatch out of the zona pellucida. So of course, to be able to grade the quality of these embryos, again, the only real thing that we have at our disposal is, of course, a microscope. So again, it's a beauty concept. Okay, we'll be looking at the morphology, more or less the similar sort of criteria for oocytes, but of course in a much more detailed manner. And ideally, what we'd be looking for is something like this. You have a nice zona pellucida outside. The cells within the zona pellucida are nice and well defined. The cytoplasm is nice and clear. So this is something called grade 1. But of course, this is the ideal scenario. Grade 1 isn't usually the one that's present most of the time. So you also have something called grade 2, where more or less, yeah, the cells look all right. You have a little bit of crap in the middle over here, but generally the cells look quite healthy. But beyond grade 1 and grade 2, you start to get other grades. And at this point, it doesn't really look like an embryo anymore. It's more like scrambled eggs at this stage. But really, the divide is grade 1 and 2, Okay, grade 1 is good, grade 2 is acceptable. Grade 3 and 4, there's just no point. Okay, this is like non-restable. The embryos are basically dying, and it's better to just discard these. So on top of these, you also have other criteria for grading of the cytoplasm of the oocyte itself. So again, this is looking at basically the clarity. Are there any indentations over here, like any craters? Are there any big holes? Are there any depressions as well? Are there any imperfections? But you're also looking at the zona pellucida. So here, is it nicely attached over here to the oocyte, or is it deformed like that? If it's deformed, then that's actually indicative of bad health. And also whether or not the pre-vitelline space, i.e. the space between the oocyte as well as zona pellucida, is that nice and clear, or does it have lots of debris floating around, such as in this example? So it's basically, again, a beauty contest. So in a nutshell, that's essentially what we have, okay? These are the basic principles of ART. You need to isolate your sperm, your sperm and your oocytes, you need to identify the best ones. You need to combine the best ones using the appropriate technologies. And then you will generate embryos, and then you will select the best quality embryos and transfer them back into the uterus. And beyond this point, actually, people would say, yeah, there's many different technologies that we use to ensure this. There's also drug treatments, etc. But beyond this point, the only real thing that we can do is basically just sit down and make dua, because after that, it's still very much 50-50 whether or not these embryos will even implant. And that brings me on to certain issues that are associated with ART. Okay. Now, I spent around about what is it now, about 35-40 minutes talking to you about how amazing and how cool this technology is, and how widely it's used and how successful it is as well. And generally that's true. But at the same time, that doesn't mean it's some sort of silver bullet. That doesn't mean that it's completely perfect. There are, of course, a lot of issues that are associated with it. The most common of these is that, not anyone can do it. You need highly trained individuals to be able to perform these highly specialized set of techniques. Okay, so what you see in here, this user over here, he or she is responsible not just for performing the technique, so it's an embryologist or an andrologist that is doing this, but this person also basically needs to be a technologist. They need to be able to have a basic command of all of the microscopic features that are over here in case anything goes wrong. So it requires extended training. But really, that is an issue that we are rectifying at the moment. The real issue is that even though ART can be used to treat around about 70% of cases of infertility, the success rate isn't very high. The success rate being successful fertilization and indeed successful pregnancy. Now, globally, very generous estimates suggest that these success rates are around about 30% per cycle. Now, one single cycle being a round of diagnosis, referral to the clinic, hormonal treatment, after that, gamete isolation, combination, selection of embryo, and then reintroduction into the uterus. So that is one cycle. And most of the time, the success rates rarely exceed 30%. In fact, for IVF and ICSI specifically, it's around about 22 to 25%. So there's a lot of room for improvement, okay? And a large part of this is that, there's a lack of global standardization in terms of techniques and criteria, but generally these technologies really need to be improved, because at the moment, even following ICSI, where you're taking a sperm and injecting it directly into the oocyte, even that isn't resulting in anything. So there's something that we're completely missing over here. And there's lots of research actually that's going on at the moment. But apart from that, I just also wanted to touch upon other applications of ART. So it's not all doom and gloom, but because there are issues that are associated with ART, these are also hindering advancement in other fields. Now, one would say, well, okay, assisted reproductive technologies, it's really used for IVFs. What type of other technologies are you talking about? Well, recently, such technology has been used to treat the incidence of mitochondrial disease. Now, mitochondria, as most of you will know, the majority of mitochondria in an individual will be inherited from the individual's mother, i.e. the oocyte. Okay, so the sperm also has mitochondria, but it's a minuscule amount compared to what the oocyte confers upon the embryo. So here you can see mitochondria stained within red within a developing oocyte, and by the M2 stage, which is the mature stage, a mature oocyte, you can see that the sheer amount of mitochondria that are present and scattered around throughout the cytoplasm. Now, what you can get is if the mother has a very large load of mitochondrial disease, so any mutation, as you know, mitochondria has its own unique set of DNA. You have a mitochondrial genome. So these are specific genes that are involved in very important processes. And of course, you will get your mitochondria, the majority of your mitochondria from your mother. So if your mother is affected with mutations in your mitochondrial DNA, this will, of course, affect you. So ART is actually being used to rectify these conditions. Okay, and then really what's being happened is using say you have a husband and wife, but the wife has abnormal mitochondrial DNA. What ART is being used to do is using IVF or ICSI sperm. The husband's sperm is combined with the wife's oocyte. So genetically, the new individual that's being formed, the pronuclea, this genetically belongs to the husband and wife. So what will happen is that this DNA, this combined DNA, will be removed from this oocyte. And then using a donor oocyte, so person number 3, female actually, where the DNA has already been removed, this couple's DNA will be introduced back into a donor oocyte. So this does not have any of the DNA of the donor. The only thing that is being used, or the only thing from the donor that would be here, would be the mitochondrial DNA, which would be normal. Okay, so essentially, this the problematic mitochondrial DNA is being removed altogether by utilizing a donated oocyte, and of course, you will then get embryogenesis. So genetically, genomically, this baby, this embryo, would still belong to the husband and wife, but the oocyte, the cell, would be from a different mother. And this might sound very controversial, and of course it is. It's being talked about in terms of the media as 3-parent babies. Now, technically, that's not really true, because the mitochondrial DNA, of course, you could get mitochondria being donated at any specific stage. But really, there are 3 individuals that are being involved. And yeah, it might sound quite controversial, might sound like some sort of space age technology, but it's actually being done. In fact, recently, I believe, I forget specifically which date, but within the past 5 years, this technique was performed for the first time. And actually, the way in which it was performed is actually quite indicative of technology at the moment. So the actual technology was performed in Mexico using an American couple with a British medical team that was performing it. So very international science. And of course, resulted in a successful individual. And this baby, I'm happy to say, thus far is completely mitochondrially normal, and no other issues seem to be plaguing this baby as well. However, this is assuming that ART itself is not causing any diseases. Here, you see, I put this picture up. You have Sir Robert Edwards. Again, unfortunately, shortly before his death, you have Louise Brown, her mother over here. Here you see Louise Brown's first son. And I put also this picture up because Louise is pregnant with her second child in this picture. So it is, again, pretty poignant. Now, the reason I put this actual picture on is that ART has only really been around for 1, maybe 2 generations, being applied first in the early 70s. So all of the babies that were created using ART are only just growing up and having children of their own. So what this really means is that we don't know whether ART is exerting any cross-generational effects. Is ART causing any issues in the children of the children that it created? Are there any developmental defects? Are there any epigenetic non-inherited defects? Now, the reason why I'm bringing this up is actually a lot of data is suggesting that there might actually be. Specifically, epigenetic diseases. So here you have something called Beckwith-Wiedemann syndrome. You also have Angelman syndrome, as well as Prader-Willi syndrome. Now, you don't, for this purposes of this lecture, you don't need to know the details of these. You don't need to know too much detail in these as well. What you do need to know, though, is that all of these are epigenetic conditions. Whereas you have epigenetic modifications of the DNA, which leads to these particular conditions. Now, the confusing thing here is that it's not that ART has directly been shown to cause these particular conditions, especially Beckwith-Wiedemann and Prader-Willi syndrome. Well, what we've just noticed is that compared to the general population, a larger portion of ART babies seem to have these particular conditions. Now, is this because ART or any step along the way of ART is causing these conditions? We don't really know. Is the ART itself posing a greater risk? Well, we don't really know. There is, however, this risk that is present. And there's a lot of body of research which is actually investigating this. But it's not just epigenetic conditions as well. There also seems to be a long-term effect of ART on genetics itself. So specifically, a lot of studies, this one specifically is showing that in babies that were born via ICSI, this is what this study was looking at, that compared to normal children, ICSI babies had a poorer quality of sperm. So in terms of sperm concentration, in total sperm count, total motility, the sperm from ICSI babies was significantly lower compared to their normal counterparts. Now, one would suggest that, well, okay, considering that ICSI was performed because there was perhaps a condition in the father to begin with, so, this genetic condition would then be passed on to the next generation. The issue, though, is that these conditions, which would normally, by just natural selection, people with genetic conditions would not be able to have children, where naturally, quote unquote, they would be taken out from the gene pool. These conditions are actually being retained. So is this some sort of compounding effect that these, because of such technologies, these genetic conditions are not just being passed on, but perhaps being amplified within the population as well, particularly considering the increasing proportion of the population that is being born via ART. But more worryingly, though, what we found is that in this recent study back in 2017, and there have been a lot more since then, is that in the past 60 years, sperm from males, albeit in fertility clinics, but males from Western populations, so developed world populations, US, UK, Europe, Australia, so on and so forth. Over the past 60 years, there has been an approximate 60% decline in male parameters, specifically male fertility. So get this, essentially, this is what this data is worryingly showing, that over the past 60 years, male parameters have fallen, okay? So what does this data suggest? Does that mean that more and more people are reporting to the clinic with fertility issues? Or does it actually mean what it says, that that humanity or human males are becoming more and more infertile as time goes along? Actually, we don't really know. But there are these issues that are associated with it. Okay. Now, just to kind of conclude, look, whether you like it or not, whether you agree with it or not, whether you think it's ethical or unethical or whatever, the fact of the matter is ART is here to stay. It's successful. It can treat infertility. Okay. One of the biggest issues that are being associated with humanity at the moment, of course, we're all preoccupied with the current pandemic situation, but this really is, the infertility is a ticking time bomb. Fertility issues are increasing. Males seem to be becoming more infertile. These technologies are going to be applied more and more in the future. Now, whether this is good or bad, we don't really know. But instead of taking a back seat and saying, oh, I disagree with this and we shouldn't be doing it, I believe that the way forward is to take an active role in this, to try and improve this technology, understand this technology as well, and perhaps try and improve people's lives as well. So I'm going to leave it there. Thank you very much for listening, listening to the ranting, basically. I think we have a little bit of time left now for questions. So does anyone have any questions? Go ahead in the chat.
Doctor?
Yes, go ahead.
Okay, so you mentioned about ART, the problems with ART and etc., right? You only mentioned that there were problems with the sperm, like as in sperm infertility and sperm problem that causes all that stuff. Does it also occur with the eggs or just the sperm?
So yes, because of course we have limited time here, I wanted to use, not, you see, this is where you have to be careful. There have been things which have been associated with both gametes, of course, okay? So the reason I use the sperm was because there's a lot more data on the sperm, because studying sperm is, as you might imagine, a lot easier since we have a lot more cells that are being produced. However, there are issues, of course, that are associated with female infertility. You have chromosomal segregation issues. You have developmental issues as well. So in fact, most epigenetic conditions seem to be attributed towards an insufficiency within the oocyte itself, or rather the environment that the embryo is implanting in. You have to remember that the very first, let's say, week to 10 days of the embryo is spent outside of the female environment. And once it's put back into the uterus, it might be incompatible. There might be other issues that are being produced there. So yes, there are a number of issues, but in terms of the mountain of proof, it's not really there, not because it doesn't exist, but because it's much harder to study, let's say, 8 cells compared to 5 million cells. Does that make sense?
Yeah. Thank you, doctor.
Any other questions? No other questions? Well, okay, wonderful. So at this point, then I'll leave it. Give you five minutes to recover for the next lecture. If you do have any questions, feel free to just contact me via email. If you find the time, come and see me as well in my office. I'm BG85, and I'll be happy to answer any questions. So thank you very much, everyone. Hope you have a really good day ahead. Take care.